Welcome everyone to this exciting global webinar on clinical metabolomics. I'm Eli Fuchs, R&D Manager at Roche Diagnostics and Chair of the IFCC Working Group on Metabolomics, and I'll be your moderator today. This webinar is an initiative of the IFCC Working Group on Metabolomics and part of our mission to improve the understanding of the technology and to make educational materials widely available to support clinical researchers, laboratory professionals. The IFCC Working Group is dedicating to evaluating cutting-edge metabolomics approaches, identifying barriers to clinical implementation, and accelerating the translation of groundbreaking discoveries from research into real-world diagnostic and patient care. So before we get started, I would like to present the IFCC Working Group on Metabolomics. We came together to explore the latest in metabolomics technology and understand what is preventing these innovations from making their way into clinical practice. Our main goal is to bridge the gap between cutting-edge research and everyday diagnostics, helping to improve patient care worldwide. I believe that what really makes us very strong as a group is our diversity. We have members from academia, hospitals, private labs, and IVD industry, bringing a mix of expertise and perspectives. Our team spans across Europe, Asia, and North America, which helps us approach challenges from all angles and really understand what's needed in the field of clinical metabolomics. Today, we are thrilled to have three international recognized speakers with us, who are also a member of the working group: Professor David Fidecchi, who is a leading name in clinical mass spectrometry, Professor Corrado Di Natale, well known for his groundbreaking work in volatilomics, and Dr. Marie Lenski, who has made a mark in clinical and forensic toxicology. Each of them bring unique insight and experiences, and I can't wait them to share their knowledge with all of you today. But before we dive into our speaker's presentation, I would like to show you only 2 slides to put clinical metabolomics into a global context, highlighting its current landscape and its potential for new applications. The working group conducted a global survey to gain insight into the state of clinical metabolomics. The survey collected responses from 400 professionals across 79 countries, highlighting the strong interest in clinical metabolomics. 43% of the participants reported that they have or are using metabolomics for clinical application. The results of this survey are crucial for understanding why we need a global context for metabolomics. They underscore the importance of this field addressing unmet clinical needs and highlight the potential for discovering new applications in the clinic. We published the results of this survey in CCLM and hope that it provides a comprehensive overview on how diverse the applications of metabolomics really are and how much more potential lies ahead. This slide highlights the current applications of metabolomics in clinical practice and offers insight into future possibilities. As we anticipated, the majority of the professionals who responded to our survey are using metabolomics primarily for research purposes. However, what stood out to us was the fact that 61% of participants are already applying metabolomics for clinical diagnostics, 42% for patient monitoring, and 36% for prognostic purposes. Moreover, what you don't see on that slide, but that nearly half of the participants expect a significant growth in the application of clinical metabolomics over the next 5 years, particularly in the field of metabolic diseases, oncology, and toxicology. So these findings emphasize the potential for expanding the role of metabolomics in clinical settings, indicating a promising future for its integration into routine medical practice. So with this context in mind, I'm excited to introduce our first speaker, David Fidecchi. So David holds a PhD in medical chemistry and biochemistry and is a professor at Palacký University in Olomouc in Czech Republic, where he leads the clinical mass spec group. With over 120 scientific publications, he has made a significant contributions to the study of inborn error of metabolics. This field is crucial within the healthcare system, as early and accurate diagnosis can lead to timely intervention, ultimately improving patient outcomes. David, the stage is yours.
Thank you very much. Do you hear me, please?
Yes.
Okay. Again, thank you very much for the opportunity to show you how metabolomics can be useful in inverse of metabolism, in diagnosing or treatment, and generally helping doctors in this area. On the first slide, let me introduce what means clinical metabolomics. It's the last step in omics, from genomics to proteomics up to small molecules, as metabolomics area, which are closely influenced by many environment, lifestyle, and other. Official term is like a field of life science research which use high throughput technologies, mostly mass spectrometry, to identify and characterize all small molecules in given biological systems, which means like biofluids or tissues or other biological systems. Generally, we can use two methodological approaches. The first one is called targeted metabolomics, and it's based on selected set of metabolites. Mostly we are using something between 100 and up to maybe 300 metabolites. And it's really useful for understanding of pathobiochemistry. And in last years, it's going to routine diagnostics processes and methods. The second approach is called untargeted, and we can know it also as a profiling metabolomics, and it's mostly used in discovery phase to find new biomarkers, and if you're lucky, also to find new disorders. The untargeted approach uses like features, so they are not analytes or metabolites or intermediates of metabolic pathways, but just the potential analytes. And by this approach, we can identify more than 1,000 features. If you will go to Medline or PubMed, you can see more than 28,000 scientific articles focused on clinical metabolomics. The question is why metabolomics is not used routinely in laboratories, I think in diagnostic laboratories and in hospitals. And the answer is that it's really complex process, and it contains, or it consists of a really complex workflow. You have to imagine that we are trying to analyze, identify, detect, and finally quantify hundreds up to 1,000 metabolites or analytes, and it requires many specific steps which are displayed on this slide, like just like an overview. And there is the common parts like the pre-analytics, analytics, and post-analytical phases. If you will go, for example, to scientific articles, you can find many metabolomics projects. And what is the reality, or what is the common in these projects? There's like a start of this project and the end, and it covers all these steps, what you can see on this slide. But in our diagnostic process, it's like never ending. So this is not fully compatible with scientific projects, and we have to add like a compatibility batch to batch and long-term applicability of metabolomics. So we have many difficulties to apply metabolomics and lipidomics workflow to routine diagnostic processes. The question why inverters of metabolism, here's the answer on this slide, is the second mostly mentioned in survey that is used in laboratories around the world, and after the metabolic diseases. And if I will go to history, the mass spectrometry and organic acids by mass spectrometry were established in 1960s, and even before starting of metabolomics as the field, as the scientific field, we used really similar analytical methods and techniques which are today applicable even in metabolomics. So therefore, inverse of metabolism is attractive field and is really close to metabolomics. What is it as inverse of metabolism? This is a huge group of diseases, today close to 2,000 different diseases. All of them are monogenic diseases, and generally we can say as enzymopathies. They are characterized by abnormal levels of metabolites in biofluids, and analytical methods are mostly used for diagnosing and for monitoring of these big groups of disorders. On the next two slides, I will show you two examples of successful application of untargeted metabolomics. The first one is from our laboratory, what we applied on beta oxidation defect. It's medium chain acylcoenzymate dehydrogenase deficiency. The shortcut is MCADD. And we applied a common liquid chromatography together with high-res mass spectrometry technology to measure more than 600 potential metabolites as features. And we found really a nice discrimination of dry blood spots from patients against the healthy controls. You can see in the middle, in the table, that there are significant features which are mostly discriminating these two groups. And you can see specific carnitines, octanoyl carnitine, decenoyl carnitine, and hexanoyl carnitine, which are mostly used for diagnosing and for numerous screening. But the first one was oxidative phosphatidylcholine as a oxylipin or oxidatively oxidized lipid, sorry. And finally, we found pathobiochemical mechanism as oxidative stress in the patients with this MCADD or MCAD disorder. And it was really useful that, or the first finding in this group of metabolic disorders, and in next years it was confirmed in the neighbor in long chain acylcoenzymate dehydrogenase deficiency. And this was first part of this project. It was like discovery, 25 controls against 25 patients. And on right side, on upper side, you can see ROC diagrams, which are really perfect or close to perfect, with AUC 0.96 or even more. But after this phase, we should go to validate these results. And we took 200 healthy controls and the same patients. And we were surprised that the clinical effectivity is not so perfect. You can see decrease to 0.93 or 0.84. And this is reality of many metabolomic projects that if they are tested on selected cohorts, they are mostly perfect. But if you are going to real life, we have many difficulties to apply it and to have it as specific biomarkers. So it was first project, or one of the first projects focused on untargeted metabolomics in our laboratory. It was published in 2015. And the second one is from Nijmegen laboratory in Netherlands. And I think it's really clever workflow how to apply untargeted metabolomics in diagnostic process for diagnosing of inverse of metabolism. The colleagues are using high-resolution mass spectrometry as a technology, and they are using two ways. The first one is in the middle in this graph, in this scheme, and it's IEM panel analysis of targeted metabolites. I think there are more than 300 selected metabolites and biomarkers. And on the right side is called Open the Metabolome. And they are using the untargeted capability of this technology to find unexpected new metabolites, new features, which can be related to unexpected disorders or even new diseases. The colleagues use the term next generation metabolic screening for this workflow. On the right side, you can see adoption or inclusion of these results to metabolic pathway and to decision process. There is, for example, xanthinuria as disorder, where we can see the high level of xanthine and decreased level of uric acid in serum. On the next two examples, I will show you application of targeted approaches. The first one is the method, what you will see on next slide, and it's finally based on our experience and our knowledge of targeted metabolomics, what we started to develop in 2010. We used the method what was published, I think 2006 by Bayard Group, and we improved the number of metabolites up to or more than 300. And we are using until today this method for targeted metabolomic projects, I think scientific projects. But during the next 2 years, we looked at this method that it would be really useful for diagnosing process if we will select just the biomarkers from this big list of metabolites. And the method what we developed was published this year, finally. So it took maybe more than 10 years to finish this work. And we apply this targeted metabolomic approach and the same conditions for separation and for detection by mass spectrometry. And we included 65 analyses to this analysis and to this method. It's really fast because we just can use the urine just to dilute to 1 millimole per liter of creatinine to add internal standards and analyze. And alone analysis takes less than 10 minutes. We are trying to find a robust solution for long-term application and long-term using of this method in diagnostic process, in the routine diagnostic process. And we found so-called robust standardized value as a scaling method for like scaling between the many metabolites, which has really different concentration and different levels. Finally, what was really useful for us was visualization by advanced methods. On lower part, you can see the cytoscape freeware, where we developed or created a metabolic network of disorders related to biomarkers. And on this slide, you can see the patient with amino acylase deficiency, and you can see specifically increased acyl amino acids. You can see acylserine, acetylleucine, and other acetyl amino acids. And it is easily visualized in a metabolic network, and we can see during a few seconds where is the place, where is this disorder, and the abnormal metabolites on left bottom part of this network. Finally, we tested this disorder even in 3 brothers, and here you can see for each brother different color of specific biomarkers, and you can see similar levels or similar increasing of acetyl amino acids. What was surprising for us was that just one of these brothers suffered from clinical presentation of this disorder. The others 2 were without clinical presentation. And the last targeted approach, what I'd like to show you, it's like new or like a changing of GCMS method for organic acidurias. I think majority of laboratories which are doing diagnosing of inverters of metabolism, are using also GCMS for organic acidureas, for organic acid measurement in urines. It's really time-consuming method, even in sample preparation and analysis and data evaluation, and it takes long hours to have results for doctors. And after our experience with targeted metabolomics based on reversed phase chromatography, we try to apply this method or this approach to organic acids measurement, and even more, we try to develop so-called semi-automatic workflow to be fast as possible. So the sample pad is the same like in the previous method, just dilution to 1 millimolar creatinine, addition of internal standards, even without centrifugation. So after that, analyzing approximately 20 minutes, and we're able to analyze in one analysis 100, close to 150 metabolites as biomarkers, as diagnostically important compounds. After that, we used automatic data processing and again, robust standardized value calculation and visualization by the same tools as in the previous paper. We did the full analytical validation and full clinical validation on this of this method, and even we went to really deep clinical validation during the 2 years. More than 1,500 urine samples and close to 100 1,500 samples of healthy controls and close to 100 samples of patients with more than 30 different disorders. This is the separation, how it looks. It's really complex, and you should combine all these 4 graphs together because it's just one analysis of all standards. And you can see many carboxylic acids, many oxo, or just not so many oxo acids, and acylglycine and acylcarnitines as confirmatory biomarkers for findings in organic acidureas. Even we are able to include selected heterocyclic and containing acids. On next slide, you can see the separation of isomery compounds like hydroxybutyrates, and we're able to separate all of them for a few of them just based on separation, and the rest of them based on mass spectrometry specific conditions. How to present results? The most easy is to use GraphPad software for us, I think for us, to use GraphPad software, and on these, just one slide, you can see all these 150 metabolites in these 2 plots, and you can see strongly elevated compounds, for example, on the right side, like phenylpropyl glycine and other specific compounds for medium chain acid coenzyme and dehydrogenase deficiency. The gray dots are healthy controls, and the red one is just one patient for all these metabolites. If I will go in more detail, you can see, for example, the sample from external quality control of the same disorder, and you can see, again, increase the specific biomarkers for MCADD. Similarly to previous paper, we established a metabolic network specific for 80 different disorders, and again, for easy visualization of abnormal findings. And in detail, you can see here from MCADD with a huge elevation of phenylpropyl glycine. And the last 2 slides are just showing us that we were able fully clinically validate these methods and these results. We were 100% succeeded in external quality control programs. And what is the biggest advantages for us is the speed of results. We are able to offer the results within 1 hour after obtaining the sample. And today we can say that it's really robust over years to use in a routine diagnostic process. The method has also limitations like oxalate difficulties and unstable oxoacids and other kind. So finally, big thanks for all of my friends and my colleagues. And on last slide, let me invite you to Metabolomics 2025. If you are interested to this field, please welcome and let's go to the Prague. And thank you very much for your attention. So that's all.
Great. Great. Thank you, David. I'm back here. So thanks a lot. That was an impressive talk. Targeted assay with many different parameters, the different compounds. So that's quite impressive. So thank you very much. I would like to mention also that if you have some questions during the talk of our speakers, please, you can submit your questions directly on the platform. After the last presentation, we will go through those questions. So thank you. So without further ado, now next we have Professor Corrado Di Natale, who is a distinguished expert in volatilomics. He earned his PhD in electronic and engineering, and he currently teaches at the University of Rome in Italy. He has authored more than 500 papers and is known for his innovative work on analyzing volatile organic compounds for non-invasive disease detection. This area is becoming increasingly important in the healthcare system as it offers less invasive methods for early diagnosis, reducing patient discomfort and healthcare costs. So, Corrado, please go ahead. Okay. Did we lose Corrado? Is there? Corrado, you might want to unmute yourself and allow the camera again.
Okay.
Great. And we can't see you yet?
Yeah.
You can activate your camera again, and then we're good to go.
It's okay?
We still can't see you, but I can hear
you. I'm on.
All right. Okay, then.
Okay. You
go
get
ahead.
it?
Thank you.
Okay. Thank you. Thank you, Eli, for the introduction, and good afternoon, everybody. And I'm going to talk today about the diagnostic properties of human volatilome. The objective is just to give an overview of the possibility that the detection, the measurement of human released volatile compounds can offer. And this is the outline of what I'm going to say. I will start to introduce the volatilomics, what we do expect, what do we find in a human volatilome. And then I will give, let's say, some example of targeted and untargeted volatilomics. And I will finish to introducing the opportunity that the transition from the classical analytical chemistry to the microelectronic sensors can offer. And volatilomics, let's say, takes into account just the smallest molecules of metabolites. We know that metabolomic deals with the small molecules, but the volatile compounds as the smallest of the small. So in volatile compounds are, let's say, compounds which are small enough to be present, let's say, in the atmosphere, in the air, and these are usually volatile compounds, as I said. So at the standard temperature and pressure, these compounds are actually liquid, but due to phase equilibrium, there is a portion of these molecules in the air. It's interesting to detect the concentration in the air because there is, let's say, a correlation between the concentration in air and what is found in liquid. So it is, let's say, a worth investigation, let's say, to measure the volatile compounds. So what are, let's say, the volatile compounds? What do we find and why we found the volatile compounds? Of course, these molecules are released at the interface between the body and the external environment. Some of these interfaces, let's say, are quite obvious. For instance, breath or skin or let's say, sweat, let's say, saliva. In other cases, the volatile compounds are captured as the headspace of human samples, for instance, urines or blood or feces. So the main, let's say, distribution of metabolites along the body fluid until to reach the external atmosphere is usually blood and lymph. So these molecules travels in blood and lymph, and they are exchanged in the air at the different interfaces. For instance, at the at the blood-air interfaces in the lung, or the blood-water interface in the kidney. So it's easy to understand that due to the different physical chemical properties of molecules, the quantity and the quality of compounds that we find in the different samples is different. And how many compounds we have, let's say. Now a recent review, let's say, collected from different literature more than 4,500 compounds, differently distributed. Breath is the most, where we find the largest abundance of compounds. And even, as I said, the quality is different. For instance, in breath, we have an excess of hydrocarbons, but in urine, due to the different solubility of these molecules, this concentration is smallest, but we have abundance of, for instance, ketones. And this means that measuring the volatilome also depends on what kind of sample is investigated. The very interesting, the first, let's say, attractiveness of volatilome measurement is the supposed simplicity, because, for instance, in breath, of course, they are released with the breath, so does not need additional, let's say, painful or whatever sampling. But nonetheless, there is, let's say, it is not simple. There is a lot of, let's say, of details that have to be considered when the volatilome is sampled. Another, let's say, important consideration about breath is that the interesting part is, let's say, very marginal respect to the total abundance of molecules that we find in breath. About 1% are volatile organic compounds. The others are the air that we breathe. Water and CO2, of course, are also very important, let's say, element. This means that the analytical instrument has to be accurate and resolute in order to capture this little amount of compounds. As we have listened the previous lecture, in metabolomics is customary to define targeted and untargeted approaches, and even, of course, in volatilomics we have, let's say, these different methodologies. And of course, in targeted, we know what we are looking for because there is a pre-existing, let's say, hypothesis or pre-existing, let's say, inference, and we know that we have to look from a specific compound. So here it is important to consider the machine that is used for this scope. Analytical machines, the measure of volatile compounds is a typical task of analytical chemistry, and it is important in many other practical applications, food, for instance. And there is a lot of machines that have been developed for the scope. All this machine basically works on the principle that they separate, distillate the compound. We have to imagine the sample as a mixture of different compounds, and we have to distillate them in order to detect individually. And this can be done, for instance, in a chromatographic column, according to the affinity that each molecule has with the solid phase on the wall of the column. But this can also be done ionizing the molecules, for instance, adding a positive charge as in a proton reaction transfer machines, and then to separate the molecule according to the mass to charge volume, since the charge is the same according to their mass. So there is this separation that is important. In some cases, like in GC, the separation takes time, and the typical chromatogram has this shape. We have in time, at a different time, appears the peak related to the compounds. In other cases, like mass spectrometers, we have real-time detection because the ionization takes place together for all compounds. So when we are interested to the target, we have just to find in this forest of peaks those that are interesting, to quantify them with using standards, and then to use, let's say, this information. In case of untargeted, we start without hypothesis. Let's say there is not a general hypothesis that fixes which compound are interesting or not. So we are going to generate relationships. It is possible also to identify, because some machine provide a sort of a fingerprint of each compound. So comparing with libraries, this can be done. There is also a different, let's say, way of treatment data, because if you are looking to simple individual compounds, the data analysis is simple. But if we look to the complex of them, we are just analyzing patterns, and then we have to use multivariate analysis, multivariate statistical analysis, what is now called machine learning. In case of targeted volatilomics, the amount of volatile compounds that we know it's important to detect may be very limited. And we have just some example of compounds that we can correlate with a specific disease. For instance, we know acetone for diabetes, ammonia for kidney, even nitric oxide is not a volatile compound, it's a gas, but is that detected in breath is important, for instance, for asthma. And there are positive correlation, and then there is attention towards this compound. However, the concentration of these compounds does not correlate with a single pathology. So this makes, let's say, we cannot consider them as a real marker for the pathology. Nonetheless, they have, of course, are very interesting, and there is a lot of studies about the detection of these compounds. Another important, let's say, approach to targeted volatilomics is when the metabolites can be induced. This is, for instance, the example in this application, in this study, where, let's say, which is aimed at studying, predicting, let's say, the concentration of a therapy or valproic acid in case for epileptic patients in serum, just measuring the volatile metabolites of the drug. Here we see one of these real-time mass spectrometers where the patient can directly blow into the machine, and from the metabolites of the valproic acid, using, let's say, a proper algorithm, we can predict the concentration of the drug in serum, and then derive, let's say, information about the efficiency of the therapy. In case, let's say, the untargeted volatilomics has a completely different approach. As I said, this is just an example. And here, the example was just, let's say, considering a cohort of patients affected by lung cancer. And these patients, let's say, are compared with controls. This data had been taken during a massive screening of population at the Istituto Europeo Oncologico in Milano. So we have a a forest of different compounds. And of course, we have just to move inside this forest. We can make a selection, can select, for instance, a subset of compounds that can be, let's say, that show a larger correlation, identification, let's say, of the disease respect to the control, and then you use the pattern of signals related to these compounds to train a classifier, a machine learning algorithm, and we can reach, let's say, a no bad, let's say, concentration. No bad performance in terms of true positive rate and true negative rate. What is interesting here is that these compounds are not specific for lung cancer, but if we look at the literature, we find that some of these compounds can also find in other kind of cancer, but even in other kind of diseases. This is just a very specific of these studies, that it is not the individual compound that can provide information on the disease, but rather its pattern. So how it is, the companions that at which he shows, let's say, in our sample. Another, let's say, application that I like to mention is offered, let's say, by the improved technology, analytical technology, the so-called ambient ionization. In this case, we collected the urines, so you brought the urines to the lab and did the measurement, so in a very standard way. In the ambient ionization mass spectroscopy, it is possible to measure directly the volatile compounds on site. And these resulted in a machine that could be used, for instance, in a surgery theater. So in a vivo or in the excised tissue can just analyze and see if the tissue is cancerous or not. There are a number of papers that this group, let's say, provided in time in this last year. So the technological advances then can really bring this volatile study very close to the practice. And in the rest of the talk, I like to talk of something that I know better, the opportunities offered by sensors. We know how sensors are changing our life. If we think how many sensors we have in our smartphone and how powerful these sensors are, we can measure acceleration, pressure, gyroscopes, and so on. And so it is very important, let's say, leap of technology to be considered. Which kind of benefit it brings? Of course, all the benefits of microelectronics, miniaturization, multiplicity of devices, rapid connection with the communication infrastructures. So all these benefits have, of course, to be connected with the chemical information that these sensors are actually able to capture. In real life, we have, for instance, the biosensor used to detect glucose, and we have been during the year how these sensors became more and more integrated with the current microelectronic devices. And the concept of making these sensors wearable is very alive, let's say, is prompting a lot of researches. So what we can do with sensors, in case we are interested to measure a specific target, we should develop a specific sensor. So a device that is absolutely selective for one compound. This is very difficult and can be done only in few particular cases for volatile compounds. This is, for instance, the example of acetone. In case of acetone, have been developed sensors that are quite sensitive and also selective to acetone. And here we see the relationship between the sensor, let's say, predicted concentration in breath respect to the concentration measured with an analytical machine. These sensors are very small, and these sensors can also be integrated, let's say, with the microelectronic infrastructure, for instance, Bluetooth and so on, can be implemented, for instance, in face masks, so can give, let's say, the reaction of the subject, even to different stimuli, diets, the relationship with the food intake, but even the physical activity and so on. Completely different is the story when we consider the untargeted volatilomics, because if we follow the same principle, we should develop one sensor for each compound that is present in the volatilome. And I said before that there are more than 4,000. So how we can do this with devices? We cannot think to develop thousands of devices absolutely specific for each compound. And here we get, let's say, inspiration from the nature, and this is, let's say, what is called artificial olfaction. Probably we know, everybody knows this. And in artificial olfaction, we try to exploit the fact that in a human, but this is common to all animals, olfaction, we have a limited number of receptors, for instance, a little bit more than 300 in human, but with this limited number, we can measure many compounds. We can detect, of course, somebody calculated trillions of different odors. This is probably too much, but nonetheless, surely more than 300. Which is the principle, the so-called combinatorial selectivity. So this means that the same compound is sensed by more receptors, and the same receptor can sense more compounds. So what makes the identification is the pattern of responses. And we try to implement this concept with the sensors. In order to do this, we need a very simple way to make sensors different one each other. Let's say, because the combinatorial selectivity relies on the fact that each sensor is different from the other. No specific, they sense more compounds, but each in a different way. And so I show you a couple of examples. This is from my group. And here we work with this molecule, very well known, the metalloporphyrins. They can be modified chemically in the chemical synthesis. And here we can see a couple of comparison between the array of sensors, the so-called artificial olfaction system, and the analytical system. This is for two kinds of cancer. This is the lung cancer that we have seen before, and we can replicate the performance of the analytical instrument, and the same in kidney cancer, measuring again urines, and we have a very similar performance between the volatile, the gas chromatography and the sensors. The porphyrins are very, let's say, are used also in other way. For instance, this is an example of a device that is now also on the market, and they use metalloporphyrins as a color indicator. Porphyrins are the molecule responsible of the color of the blood and even the color of the leaves. So they are very, let's say, brilliant colors. And in this case, this is a very simple. It is not a device actually, because it's a very simple piece of plastics where molecules are spotted. They are exposed to the vapors, for instance, of different bacteria, as in this case, and we measure. It is measured the differences just measuring the color with the normal digital scanner. So a machine that is has not been conceived to make a chemical or medical analysis, but was designed for a completely different scope. So this is, let's say, a very nice example about also how with sensor we can use, reuse, readapt the technology that still exists and can be used, let's say, for metabolomics studies. I'm finishing just to give you some reference. For instance, this is a very nice paper about wearable perspective about wearable. This book about, let's say, what we can find in breath, and our recent, let's say, survey and about review about volatilomic assay. Thank you very much.
Thanks, Corrado, for this excellent presentation and all those many examples where we see how this field is actually getting closer to the patient now. So thank you very much. So if you have questions for Corrado, please use the the chat for this, and we will go through all the questions after the last speaker. So finally, we are joined now by Dr. Mary Lenski, who has a PhD in laboratory medicine and works at Lille University Hospital in France. She's also a very active member of the IFCC Task Force Young Scientists, and she specializes in clinical and forensic toxicology and has published numerous papers on the application of metabolomics in these fields. Understanding toxicological impacts is vital for public health and safety, and her work contributes to improving diagnostic capabilities in both clinical and forensic settings. Mary?
Hello.
Are you here? Hello. So the stage is yours.
Thank
Thank
you.
you.
Thank you very much, Ellie, for this kind introduction, and many thanks to IFCC for allowing us to present this webinar today. It's a pleasure for me to discuss metabolomics in biological and forensic toxicology. And here the objectives of this presentation are to identify several unmet clinical needs in toxicology and illustrate current applications with examples from the literature. So first, let's introduce toxicology in the context of laboratory medicine. So toxicology focuses on the diagnosis, management, and prevention of adverse health effects due to xenobiotics exposure. So the following analytes are covered: medications, drug of abuse, metals, environmental toxins, and chemical agents. So routine toxicological workflows usually involve drug pre-screening by immunoassay, qualitative screening procedure using mass spectrometry techniques, followed by confirmatory and quantitative analysis using mass spectrometry techniques. Okay, so toxicological analyses are used in a wide range of clinical scenarios in patient care. So emergency toxicology focuses on the diagnosis and management of the critical poison patients, whether from intentional or unintentional poisoning, drugs, or medication overdose. So the key challenge here is meeting the turnaround time required for initial patient management in the emergency department. Addiction medicine is centered on the diagnosis, treatment, and recovery of individuals suffering from addiction, including substance use disorders. Occupational toxicology focuses on workers who may be exposed to toxins in their workplace. And while the scenarios mentioned are primarily encountered in hospital and workplace environment, forensic toxicology deals with judicial matters, as results are likely to be used in court. So forensic toxicology can involve living subjects, for example, regarding driving under the influence of drugs, or in drug facilitating crime. And it also includes post-mortem toxicology, which aims to evaluate the involvement or contribution of xenobiotics in the cause of or manner of death. And finally, another specialized branch of toxicology is doping control, which focuses on detection and monitoring of prohibited substances or methods used by athletes to enhance their performance in sports, to ensure fair competition in sports. As a highly dynamic and rapidly evolving field, biological and forensic toxicology are constantly confronted with both old and new challenges. So those include the need to prove acute drug consumption, extend detection windows, discriminate addiction from occasional consumers, improve post-mortem interpretation of forensic situations, or also prove unambiguous evidence of sport doping. So recently, an increasing number of publications have described both targeted and untargeted metabolomics approaches to tackle those challenges. So those experiments will be discussed in the following slides. So providing acute drug consumption for certain drug of abuse through metabolomics aims to utilize strategies that are not directly based on the chemical structure of the substance. So this includes examining the activity of a substance through a receptor activity assay, which is part of activity profiling. It also involves the identification of specific endogenous biomarkers as metabolomic fingerprint or signature. And additionally, it concerns the pharmacological profiling of drugs of abuse as a strategy to predict pharmacological profile of other drugs. So let's take the example of drug of abuse, specifically new psychoactive substances, NPS. So NPS refer to a heterogeneous range of psychoactive substances that mimic the effects of various drugs or illicit products. So many originated from scientific research and then by clandestine laboratories to evade the drug laws. The use and availability of NPS on internet has rapidly increased, and up to November 2023, more than 1,200 substances have been reported to the United Nations Office on Drugs and Crimes, with 30 being reported for the first time in 2023. So since NPS are not controlled under the International Drug Control Conventions, their legal statute can differ widely from country to country, and they pose really a major public health threat, as information on the pharmacology and the toxicology of NPS is unavailable in most cases. So on the right side of the slide, you can see a representation of the drug wheel. So the outer ring lists established psychoactive substances, while the inner ring lists newer psychoactive substances. And NPS available on the market have similar effects to those of to the substance under international control, such as cannabis, cocaine, heroin, amphetamines, and they could be classified by their effects, including stimulants, synthetic cannabinoids, hallucinogens, and synthetic opioids. So analytical challenges with NPS lie first in the variety of substances associated with a high frequency of new products, which need regular update of the analytical tool. Second, they present a low dose effect ratio and consequently low circulating concentrations, requiring the development of sensitive methods. And third, they also have a rapid metabolism, so it's essential to detect the parent drug and its metabolites to extend the detection window. So in this context, metabolomics could be a promising tool to address those analytical challenges and assist in the indirect identification of drug consumption. Some studies focuses on that idea and on metabolomics targeted approach to predict the pharmacological profile of NPS. So here the authors have characterized changes in the monoamine neurotransmitters and steroid pathway in rats after exposure to classic drugs of abuse, including MDMA, methamphetamine, cocaine, heroin, and delta-9 tetrahydrocannabinol. They have created a machine learning algorithm to predict the similarity of two NPS to classical drug of abuse. So their model successfully predict the pharmacological profile of a synthetic cannabinol, GWH18, as a cannabinoid-like drug, and also a synthetic catinone, mephedrone, as an MDMA-like psychostimulant based on the metabolomic profile, which is consistent with the literature on those NPS. So this approach allows a fast classification of NPS according to their pharmacological similarity with classic drugs of abuse, which could benefit both during risk evaluation policy and public health. Here, GHB, gamma hydrobutyric acid, is a short-chain organic acid, and it is an approved drug for treating narcolepsy. It is often used illegally for recreational purpose and also in drug-facilitated crimes by spiking GHB into alcoholic beverage. GHB is also an endogenous neurotransmitter, so the detection of GHB, and particularly differentiation between exogenous intake and endogenous base levels, remains challenging. Moreover, it is an extremely short detection window due to fast metabolism, and no metabolite of GHB has been yet identified that allows for longer detection window compared to the using of parent compound itself. So the analytical detection window are only 6 hours in blood and 12 hours in urine. So this is often too short to provide reliable proof of GHB ingestion in the context of drug-facilitated crimes. So now new strategies to demonstrate exogenous GHB administration are still of higher interest in toxicology. So here metabolomic studies were recently performed to identify endogenous markers that might be able to prolong the detection window of GHB. So in this study, the authors used the NMR-based metabolomics approach to identify time-related changes at different time points after GHB intake in a control administration study in human urine. So by comparing exposed patients to controls, the metabolites with highest influence on separation after GHB are GHB itself, glycolate and also succinate. And as demonstrated in the box plot, glycolate concentrations decrease more slowly to endogenous levels compared to GHB. So this is suggesting that glycolate could be a potential biomarker to extend the detection window of GHB intake. Now, discrimination between addiction and occasional use of drugs of abuse is an important challenge also in toxicology. So indeed, there is a lack of biomarker for addiction that could be useful in various clinical scenarios, including the diagnosis and treatment of addiction, workplace drug testing, or in post-mortem interpretation of drug concentrations. So currently, the available literature focuses on methamphetamine, cocaine, heroin, and mainly in animal models, studying exposure, withdrawal, and abuse. Metabolomic studies were recently performed in opioid use disorders. So opioid use disorder is diagnosed using the qualitative criteria defined by the Diagnostic and Statistical Manual of Mental Disorder 5th edition, DSM-5. So currently, diagnostic biomarkers for opioid use disorder do not exist. So this study focused on developing objective biological markers to differentiate chronic opiate users with opioid use disorder from chronic opiate user without opioid use disorder using untargeted metabolomics on urine samples. So here the authors found a metabolic profile of 712 peaks that differentiated users diagnosed as opioid use disorder positive. So using the metabolic profile, stepwise logistic regression modeling allowed for 95% accuracy in prediction of opioid use disorder positive patients, which suggests that metabolomics workflow could complement the positive diagnosis of opioid use disorder. Now, improving postmortem interpretation in forensic situations is also a challenge in toxicology. So during the lifespan, physiological, environmental, and pathological factors influence the expression of the metabolome. And also in post-mortem period, the metabolome undergoes time and location-dependent changes through degradation, accumulation, or redistribution of metabolites based on their physiochemical characteristics. So those changes can vary, particularly depending on the time elapsed since death, the cause of death, bacterial proliferation, environmental conditions, and other causes. So those complex multiparametric changes are difficult to fully understand with a single biomarker. So here metabolomics therefore appears to be a promising approach to improve post-mortem interpretation of forensic situations. Autopsy and postmortem investigations, such as toxicology, microbiology, and histological examination, allow the determination of the cause of death in the majority of cases. But in unclear circumstances, there is a need for novel methods to define cause of death. So here is presented a metabolomic approach applied in a pathological case of a fatal acute methadone intoxication in an infant. So this case report was initially suspected as a sudden infant death syndrome. Pathological findings were consistent with an acute respiratory failure, while toxicological analysis revealed an elevated blood methadone concentration. In addition to the routine approach, the urine sample collected at the autopsy was investigated with the NMR metabolomic approach. The identified metabolomic profile was compared with the urinary metabolomic profile previously obtained from newborns who experienced perinatal asphyxia and healthy control newborns. So the urinary profile of the methadone intoxicated infants was very similar to those belonging to the perinatal asphyxia newborns, especially to those belonging to non-surviving babies. So those data suggest a shared hypoxic signature, highlighting the potential of metabolomics as an additional tool to investigate different cause and mechanism of death. The determination of postmortem interval is also a fundamental part of forensic death investigation. So traditional postmortem interval estimation methods are mainly based on the evaluation of a single or few physical parameters that are dependent on the examiner individual evaluation and lacking to some extent precision, accuracy, and reproducibility. So metabolomics in body fluids or tissues samples may offer a more accurate way of estimating the time since death. So different animal models have already been studied, and several biological matrices such as blood, muscle, aqueous humor have been analyzed. But unfortunately, the different studies are difficult to compare because the experimental designs and conditions are different, and several confounding factors exist, such as the cause of death, selected individuals or animal, or the harvesting of the sample. I will illustrate this application of metabolomics with one example. So in this study, muscle samples were procured from 52 mice at a 7th postmortem interval from 0 to 48 hours after death. A PCA illustrated chronicle changes that can be distinguished among the 175 metabolites identified in the muscle. A postmortem interval estimation panel was then constructed with and which was constituted of 17 of the best postmortem interval biomarkers identified in the muscle, demonstrating statistically significant correlation between metabolite concentrations and postmortem interval. And here a validation study was carried out, yielding a mean accuracy of postmortem interval estimation of minus 0.27 hours with a standard deviation of 2.88 hours. So this study proves the utility of metabolomics profiling in postmortem interval estimation with biochemical profiling studies. So in conclusion, metabolomic approaches possess in general great potential as a complementary tool to those currently used in biological and forensic toxicology, as illustrated with various studies presented today. Though the scope of application are even bigger with other application detail, other unmet clinical needs, including human sport anti-doping, prediction of side effects of medication, drug monitoring, and so on. So here metabolomics is a promising approach, but several challenges must be overcome before its implementation into routine clinical practice. While many untargeted investigations have been conducted over the years, there is only few targeted follow-up studies that are currently available to confirm initial results. Moreover, the validation of the proposed biomarker for biological and forensic toxicology in human studies is now essential. Finally, standardization of protocols for sample collection, processing, and analysis is required. So there is a dire need of large population-based metabolomic study globally with a harmonized methodology so as to obtain robust and meaningful data that may be transferred from bench to bedside in clinical care. I would like to thank you very much for your attention.
Thank you, Marie. That was a very great overview on a field that's often not really known to be using metabolomics. So thank you for this. Okay, so we received a few questions through the platform. So now I'm going to invite all the speakers to switch on the camera. So we'll try to have a lively discussion for the rest of the webinar. David, you here?
Yes. Yes.
Okay, so the first question that came in was, what is the current status of the use of metabolomics in the study of patients with hereditary dyslipidemias? David, I think that you can answer this, right?
Yes, I will try. I have to say that I went to do AI and to do PubMed to look at hereditary dyslipidemia. It's really a rare disorder. Yeah, it has incidence 1 per 200,000, and there are just a few papers published in metabolomics and lipidomics. I saw less than 6, I think. So there is not known so much about this disorder related to metabolomics and lipidomics. What could be really promising is like a cardiovascular risk prognosing by lipidomics profile. So I think there is the way how we can help the patients to prevent acute cardiovascular diseases. But yeah, I can see the difficulty to have enough patients for metabolomics or lipidomics studies generally. Abby?
Okay, now I was on mute, so I will. Okay, thanks for your answer, David. I was on mute. So moving on to the second question. Could metabolomics replace conventional blood chemistry in the future in the diagnosis of pathologies? So I guess anyone can give their view on this. So it's also something we like to discuss within the working group. So maybe, David, you can give us some answer for your field of expertise, and then we'll move to Marie and Corrado.
Yeah, thank you very much. Now I can see a little bit different perspective from clinical biochemistry lab, and the answer is absolutely not. I think it will be like a piece of puzzle to be more clinically effective in diagnosing and maybe even more in prognosing, like events or like a treatment effectiveness, for example. So I think there is the way where we can be helpful. And what I can see in the last years, mainly lipidomics will help us for this reason. So This is my perspective.
Okay, thanks. Marie, would you like to give your perspective?
Yes. So I think that in the next years, it is more likely that metabolomics will complement conventional blood tests more than replace them. So metabolomics can provide a more detailed picture of certain disease and offer sometimes early detection for conditions that traditional biomarkers may miss. But there are some difficulties, I would say, due to the complexity of metabolomics, maybe the cost and the need for the validation of results that are already published. So I think that no, it will not replace classical conventional blood tests, but more complement those tests.
Okay, thank you. And maybe Corrado, your perspective more in line or in the perspective of sensors and if this can eventually play a
Well, sensors are a good promise. They make a good promise because the big promise here is just to make things simpler, to make things smaller. If we can do sensors wearable, sensors become a sort of a personal let's say, device that we can use to track the progress of diseases or even to augment the perception of our body. So there is a lot, let's say, of promises. Of course, there is a lot of work still to be done. Volatilomics is around since many years and is still in its infancy. We are, let's say, working in a kindergarten with the infants, and we are waiting. We are trying to make this infant growing. So this is, let's say, a very nice for us endeavor, and it still remains a promise. And I think this promise needs to be cultivated and to be, let's say, we have to not believe, but nonetheless to spend time, let's say, to make these promises true.
And just to add my personal view on this, it's also, I think also it's going to be directed by costs. So if there is an alternative with classical clinical chemistry, I think, which is cheaper, then of course we'll stick to this. And if there are some unmet needs that can be addressed with metabolomics, and if it's really relevant to the patients, then maybe we'll have this alternative in the future. Which brings us, so this cost question brings us to the next question. So what would be the investment? So I don't know if anyone would like to start in this. Maybe I'll give some elements of answers, but so of course the cost, you have the cost of the hardware, but you also have the cost of the experts to run those metabolomics platform and to perform data interpretation, to perform sample preparations. So it's hard to give a figure. I think they vary depending on the location as well. But you have to take into account all those components, your hardware and the staff and training, I guess.
Yeah.
Anyone else want to add some?
I agree completely. So, and what is more important is the investment on people, the investment on staff, and to maintain the staff at the level to do this research. So this is probably the great gap between the research in academy and the practical use, for it, because these people have to think in terms of continuous growing if they want to apply these techniques.
Maybe just mention that you will need not only analytical chemists, but also statisticians, and for the interpretation, also biochemists. So it's really complex task or questions at the start of these metabolomics. And yeah, to succeed, the overall workflow, it's not easy. And also this is the answer why it's not so commonly seen in laboratories. And the cost, I think, I would say, or I would answer at minimum 3 years of experience. It's even more than the cost.
Okay. Thank you, everyone. Moving on to the next question about validation, how to validate data during research work that we get from metabolomics. So data validation from metabolomics. A very important topic, actually. So would you like to go first?
Yeah, I can, Ellie.
Go ahead.
So yeah, validation in metabolomics and in metabolomics, the research is critical step to ensure accuracy and reproducibility and also relevance of results. So first, you have I will say controls that you can put inside of your batch to validate your results. So it could be based on pooled quality control samples, also blanks, also internal standards, for example. Also, it's important to reproduce the analysis to confirm the results. And also, it's important to have an external validation of biomarkers, maybe with a cross-platform comparison of results. So that's several ways that you can validate your data and your results in research in metabolomics.
Yeah, and David, you were talking about analytical validation and clinical validation. Maybe you can explain a little bit the concept of the two different types of validations.
I would say I have easy life in terms of metabolism, because we have so strong differences between the patients and healthy population, and it's really easy to discriminate in metabolomics methods. But if I will go back to my presentation, 30,000 papers was published in clinical metabolomics field. And we have no FDA approved biomarkers for clinical decision process, I think, in laboratories. And if you will go to Nature journal and try to ask for reproducibility crisis in science, you will find the answers like 85% of results are as a waste. And this is the reality. We should go back to results, to findings, and try to validate. And what I can see as a difficulty is, if we will try to find biomarkers, we should go try to validate on general population, on population of different diseases of patients. So not so easy.
So okay, so you're talking about a need for a better validation in terms of clinical validation.
Yeah.
All right, thank you. Then we have a question for Corrado. Is the urine analysis sensor system based on QCM type sensor?
Yes, the example I shown were with those sensors, but they are not limited to the urine analysis. You can analyze any volatile compounds from any origin, and I think this work for any gas sensor. Mm-hmm. So you can specialize the sensor for urines, for instance, if you have sensors that have more sensitivity towards the compounds that are expected to be found in urine. This is what makes the sensor more sensitive to a particular application. For instance, in case of urines, ketones, for instance, not hydrocarbon, so reduce that. So it's more the kind of sensitivity of the sensor than the specific technology that you use.
Okay, thank you. Next question regarding the computing part. So to what extent can the needs for high performance computing and large scale data storage be compared with the needs in genomics?
Not at those levels. Let's say we can still work on an Excel file, on an Excel table. So I mean, not that we are using Excel, but I mean something that can stay there, you know. But we do not use, of course, Excel for the analysis. But just to give an idea that what we what we collect, let's say, are still manageable data that you can manage on a personal computer. The analysis, of course, may require more sophisticated algorithms, but at the moment, I do not see, let's say, problems of computing for this kind of applications.
Yeah, I guess it's very dependent on type of technology that is used for metabolomics. So depending on the technique that's being used and whether you're talking about targeted assays or untargeted assays, they generate different levels of complexity and different dimensions of data. So for the mass spectrometry data, some data sets are quite large and are quite complex to process. And not to mention that many groups are now trying to put metabolomics in the context of other omics. So also including genomics, transcriptomics, and proteomics data. So that's also where the future is lying, I guess, where we're heading. But yeah, David, Mary, please, if you want to add something.
What's my experience? We started with untargeted methods, untargeted metabolomics maybe 10 years ago, and we were strongly limited by computation power for processing of the data. Today, what I know, it's not uncommon to have up to 1 gigabyte per one sample. If we will go to a high resolution mass spectrometry in profile mode, which contains maximum data inside, and if you try to compute it and to compare maybe 1,000 samples, it takes weeks for calculation. So it strongly, as Ali mentioned, it strongly depends on which kind of data we have, and if we will go to targeted or untargeted metabolomics.
Yeah, I totally agree with Corrado and David. It depends on the acquisition modes of data, but however, the question of data storage should be thought when you want to establish a metabolomic workflow, because it could be quickly an issue if you don't think about that. So yes, that's very important. Yeah.
Okay, thank you. So as we're running out of time, we will take the last questions before the closing remarks, and then the questions in order to get the credit for that webinar. So the last question is, at present, what instruments are available for metabolomics?
Many instruments. I talk. If you think at volatile compounds, you have, let's say, as I said, the presentation, there are two big families. Let's say the gas chromatography that you can couple with a number of different detector or even combine different columns and so on. And then the direct injection instruments, where let's say are based on the ionization of the sample. So there are many instruments, but it is important to understand that each instrument is a different point of view about the sample. So it's a different lens at which we look at the sample. If you take a gas chromatography, you have to think of the affinity of the column. If you take the mass spectrometer, you have to think which is the procedure of ionization. This means that each instrument does not provide the real composition, but it provides the composition through its lenses. So it's something different. So probably a single instrument is not necessary. It's not enough if we want to thoroughly study a problem.
To have the holistic metabolite coverage. Got it. Okay. Does anyone want to add something to this?
Yeah, if I complete what Corrado say, I would say that common technological tools that are used in metabolomics are NMR, also mass spectrometry coupled to gas chromatography or liquid chromatography. So there are other tools also, but they are the most common ones, and they are complementary tools to have a maximum coverage of the metabolome that can be expressed after pathological condition, environmental exposure, drugs exposure, and so on.
Okay. Then we are running out of time, so in order to finish at 3:30 CET now, we will close the Q&A. So there are a couple of questions for which we didn't have time to provide answers. We will try to get in touch directly with you and get you some answers. So there are only 4 or 5 questions left, but we'll work them out afterwards. So now, first thing is I would like to thank all of you for attending this webinar. It's quite exciting. Thank you, Marie. Thank you, David. Thank you, Corrado. And thank you so much for being part of this great working group on metabolomics. And that's also the opportunity to thank all the other members that I've shown on the picture. So we'd like to thank the IFCC for offering this tool for e-learning. Yeah, as I said, please remember that unanswered question will be replied by emails. The certificate of participation will be provided at the end of this webinar after a few questions. So at the end, so like now when I finish my second sentence, you can click on show my scores and get and answer the question and get your certificate. Also, be aware that you can go back to this webinar. It's recorded and will be available in a few days. You'll be informed per email. And soon there will be other IFCC free webinars, and they'll be announced via the official IFCC communication channels, or email, website, and social media. With this, thank you and goodbye.
Thank you.
Thank you. Bye.
Bye-bye.